So biosynthesis of chloramphenicol. So this biosynthesis of chloramphenicol, again it starts from chorismic acid. लेकिन यहाँ पे जो हमारा ट्रिप्टोफैन जब हमने सिंथेसाइज किया था तो chorismic acid को हमने अमीनेशन किया था by using L-glutamine. So in that case what had happened is in L-phenylalanine में हमने ट्रिप्टोफैन में बायोसिंथेसिस में हमारा सेकेंड पोजीशन पर अमीनेशन हुआ था. Now in this case instead of in the second position we can do it at the fourth position. Here we have a hydroxyl group, here we have a COOH group, and here COOH group. This is our chorismic acid. This chorismic acid undergoes amination reaction in presence of glutamine, like what we had it for ट्रिप्टोफैन. लेकिन ट्रिप्टोफैन में क्या हुआ था कि यहाँ पर अमीनेशन हुआ था. अभी यहाँ पर अमीनेशन होने की बजाय यहाँ पे अमीनेशन होगा at the fourth position, and it will be resulting into loss of hydroxyl group, and we will have amine group will be added over there. So we will get a product as NH2 here O COOH, and here we will have it. Okay. So यहाँ पर हमें ये मिलेगा, this is 4-amino 4-deoxy chorismic acid. Deoxy यहाँ से हमारा ऑक्सीजन हाइड्रोक्सिल ग्रुप चला गया. So we have a 4-amino 4-deoxy chorismic acid. Now this undergoes loss of pyruvic acid, and further loss of pyruvic acid as well as aromatization. So यहाँ से हाइड्रोजन जाएगा और यहाँ से pyruvic acid का लॉस होगा. So that we will get, it will be 1,2 cis elimination or syn elimination. So we will get this as para-amino benzoic acid. Para-amino benzoic acid, or in bracket we can have PABA. So we get a PABA PABA from this 4-amino deoxy chorismic acid. Further, if it does not undergo this loss of pyruvic acid, but it undergoes pleasant rearrangement, like what we had done it for chorismic acid to prefenic acid. chorismic acid से जो prefenic acid हमने बनाया था, उसी टाइप में यहाँ पर भी अगर हम pleasant type of rearrangement करते हैं, ये ग्रुप अगर हमारा ट्रांसफर होता है at this carbon, transfer of group from here to this carbon and this double bond coming over here, we will get a derivative of prefenic acid COOH, and here we will have a COOH, we will have a double bond here and a double bond here and NH2 over here. So prefenic acid से यहाँ पर हमें क्या है, we have a 4-deoxy 4-amino prefenic acid. This will be again a derivative of prefenic acid, 4-amino 4-deoxy prefenic acid. This we will get. Now this prefenic acid derivative then can undergo decarboxylation reaction and further aromatization. So it can undergo decarboxylation with aromatization, and mind it, here we will be retaining this amine group. So we will have a product as, यहाँ पे हमारा NH2 ग्रुप रहेगा, and here we will have COOH and here we will have NH2 group. So this we will get, this is para-amino phenyl pyruvic acid. Para-amino phenyl pyruvic acid we will get. This para-amino phenyl pyruvic acid then undergoes transamination reaction in presence of pyridoxal phosphate, that is vitamin B6. So यहाँ पर एक हमारा गड़बड़ हुआ. We have here, instead of यहाँ पर हमारा ग्रुप जो है, this we have it as a double bond O. यहाँ पर हमारा ये डबल बॉन्ड O था, वो वैसे के वैसे ही यहाँ पर रिटेन होगा डबल बॉन्ड O. So transamination reaction में it has gone down. So transamination reaction का जो प्रोडक्ट होगा, that will be वो जो कीटो ग्रुप है, उस कीटो ग्रुप की जगह पे हमारे पास अमीन ग्रुप आएगा. So we will have here as NH2 and here as COOH and here NH2. So we will have para-amino phenyl alanine. Para-amino phenyl alanine we will get. That is, we can have PAPA, as it is L, so LPAPA, that is L kappa we will get. This compound short name will be LPAPA. This undergoes further hydroxylation reaction. Hydroxylation at this position. So we will have NH2, here NH2, here we will have a OH, this side COOH. This undergoes further reaction with N-acylation reaction with CHCl2 COS-CoA, that is coenzyme A, and dichloroacetyl-CoA. So we will have, ये compounds generally नहीं मिलते हैं, particularly animals में तो definitely नहीं मिलते हैं. Plants में ये कभी-कभार special conditions में ही मिलते हैं. So we will have here as again OH, और यहाँ पे जो हमारा NH2 था, now it becomes N-acylation. So NHCO CHCl2. This undergoes further reduction of the COOH group reduction of COH group to CH2OH. We will have NH2, CH2OH, NH, CO, CH, Cl2 और यहाँ पर हमारे पास एक OH. And further, last step of this chloramphenicol biosynthesis में यहाँ पे oxidation of NH2 to NO2. ये very rare होता है. Generally NO2 groups are not present in body, in either in plants or in animals also. So oxidation यहाँ पर होता है. This oxidation we get as a last step of this to generate chloramphenicol. NH, CO, CH, Cl2, and here we will have as a OH. So this is chloramphenicol. This is chloramphenicol.